Welcome back to G Fun Facts Online. Today, we are going to talk about the most sophisticated high-voltage machinery in the known universe. It is not a particle accelerator, and it is not a nuclear submarine. It is you. Specifically, we are diving into the engine room of the human body to look at the power plants that keep you alive, the mitochondria. If you are listening to this, your heart is beating, your brain is processing my voice, and your muscles are holding you upright. All of that requires a constant, massive stream of energy in the form of a molecule called ATP. For decades, medicine looked at mitochondria like simple batteries. If the battery died, the machine stopped, and there was nothing we could do about it. But that era is ending. We are currently witnessing the dawn of mitochondrial medicine, a scientific revolution where we are moving from simply watching these power plants fail to actively repairing them at the genetic level. This is the story of how gene editing is rewriting the future of cellular energy, and it is a story that involves ancient bacteria, invisible borders, and molecular assassins. To understand why this is such a massive breakthrough, we have to start with a bit of evolutionary history. You might remember from biology class that mitochondria are the powerhouses of the cell. But they are so much more than that. They are actually aliens. Billions of years ago, mitochondria were free-living bacteria swimming around in the primordial soup. Eventually, they were engulfed or swallowed by a larger cell. But instead of being digested, they struck a deal. They formed a symbiotic relationship. The bacteria provided energy, and the host cell provided protection. This ancient pact is why you are alive today. But it left us with a very strange biological quirk. We have two separate genomes. You have your nuclear genome, the famous DNA helix that determines your eye color and height, housing over 20,000 genes. But tucked away inside your mitochondria, there is a second, tiny genome. It is a small circular loop of DNA containing just 37 genes. It is small, but it is mighty. This mitochondrial DNA, or mtDNA, encodes 13 essential proteins that build the electron transport chain, the actual machinery that generates the electricity for your life. Here's the problem. This tiny genome is incredibly vulnerable. It sits right next to the energy production site, which is like living next to a nuclear reactor. It gets damaged easily. When mutations strike this small genome, the consequences are catastrophic. We see diseases like Leber's hereditary optic neuropathy, which causes sudden blindness, or Millas syndrome, which causes stroke-like episodes and muscle failure. These diseases shut down the body's power grid. The organs that need the most energy, the brain, the heart, the muscles, they fail first. And for the longest time, these conditions were untreatable. You might ask, why couldn't we just fix them? We have gene editing tools now, right? We have CRISPR. This brings us to the Great Barrier. The 21st century gave us CRISPR-Cas9, the molecular scissors that can cut and paste DNA. It revolutionized biology. But for mitochondrial biologists, CRISPR was a total failure. It simply did not work. The reason is biology's strictest border control, the mitochondrial double membrane. This membrane is highly selective. It is designed to keep foreign genetic material out to protect that energy core. CRISPR works as a team. You have the Cas9 protein, which is the scissors, and a guide RNA, which is the map telling the scissors where to cut. Scientists could figure out how to smuggle the protein in, but they could never get the guide RNA across that double membrane. Without the map, the scissors were useless. So for nearly a decade, while the rest of the genetic world was celebrating CRISPR, mitochondrial editing was considered impossible. The field needed a new approach, one that didn't rely on RNA. The solution came from revisiting older technologies and merging them with brand new discoveries. If RNA couldn't cross the border, scientists realized they needed fully protein-based editors that carried their own targeting instructions. Essentially, a robot that didn't need a separate map because the map was built into its body. The first breakthrough came not from repairing genes, but from destroying them. This is a fascinating concept called heteroplasmy. A single cell in your body contains hundreds of mitochondria, and each mitochondrion holds multiple copies of DNA. In a sick patient, you have a mix. Some DNA is mutant, and some is healthy. It is a battle for dominance. Scientists engineered protein-based tools called MitoTALENs and zinc finger nucleases. Think of these as molecular assassins. These proteins could be programmed to enter the mitochondria, scan the DNA, and recognize only the mutant sequence. Once they found it, they cut it. Now, mitochondria don't like broken DNA, so when the mutant DNA is cut, it is destroyed. The cell suddenly realizes its energy levels are dropping because it is losing DNA, so it signals the remaining DNA to replicate. But here's the trick. The molecular assassins only killed the bad DNA, so the cell is forced to replicate the healthy DNA. The result is a shift in the ratio. The bad DNA is depleted, the good DNA takes over, and cellular energy is restored. In 2018, this approach successfully treated mitochondrial disease in mice, proving for the first time that we could manipulate this alien genome in a living mammal. But there was still a catch. Destroying bad DNA only works if you have enough good DNA to take its place. What if a patient has 100% mutant DNA? If you destroy it all, you wipe out the cell's entire energy grid. The cell dies. We needed a way to actually fix the typo, to convert the mutation back to the healthy sequence without cutting the DNA strand. This seemed impossible because the enzymes we usually use for this require the DNA to be ripped open into single strands, something that is hard to do inside the mitochondria. Then, in 2020, a team at the Broad Institute, led by David Liu and Vamsi Mutha, shattered the dogma. They discovered a peculiar toxin produced by a bacteria called Burkholderia caenocepacia. This toxin, called DDDA, was a natural weapon. It evolved to kill competitors by mutating their DNA, but unlike other enzymes, DDDA could work on double-stranded DNA, the natural state of mitochondrial DNA. This was the key. But you can't just inject a toxin into a human cell. It would kill everything. So the researchers did something brilliant. They split the toxin into two inactive halves. They attached each half to a tail protein, our protein-based GPS. These two halves would float around harmlessly until they both landed on the exact target DNA sequence right next to each other. Only then would they reassemble into a working enzyme and perform a chemical reaction, changing a cytosine to a thymine. This was the first mitochondrial base editor. It allowed scientists to correct point mutations without cutting the DNA, making it a far safer and more precise method. The innovation didn't stop there. Just 2 years later, in 2022, researchers in South Korea developed TALEDs. They combined the DDDA technology with another engineered enzyme to achieve the ability to convert adenine to guanine. This was a massive leap. Between these two technologies, we can now theoretically target and correct nearly all types of pathogenic point mutations in the mitochondria. We have gone from impossible to mostly solvable in under 5 years. Now, you might be thinking, this sounds great in a lab, but when does it help actual people? We are no longer just in the realm of petri dishes. The race to bring these therapies to humans is accelerating, and 2025 is shaping up to be a pivotal year. One of the most watched programs is from a company called Precision Biosciences. They are developing a therapy called PBGENEPMM for primary mitochondrial myopathy. This is a condition caused by a specific mutation known as M.3243. Using their proprietary ARCUS platform, which is a distinct type of nuclease enzyme, they aim to eliminate the mutant genome in living patients. They expect to file for clinical trials in 2025. If approved, this would be the first direct mitochondrial gene editing therapy in humans. And we are seeing success in delivery too. A major hurdle has always been getting these big editing proteins into the specific organs that need them, like the heart. In early 2025, a landmark study published in Science Translational Medicine by researchers at the University of Miami, led by the Murray's lab, demonstrated something incredible. They used virus-like particles, or VLPs, to deliver a base editor to the hearts of mice. They successfully corrected a mitochondrial mutation in the heart tissue. This proved that we can not only edit the DNA, but effectively deliver the editor to a notoriously difficult organ. This brings us to the ethical landscape, which is fascinatingly complex. Mitochondrial medicine sits at a unique intersection. Before gene editing, the only way to prevent a mother from passing a mitochondrial disease to her child was something called mitochondrial replacement therapy, often called three-parent baby technology. This involves taking the nucleus of the mother's egg and placing it into a donor egg that has healthy mitochondria but had its own nucleus removed. This is legal in places like the UK and Australia, but it remains controversial because it modifies the germline, meaning the changes are passed down to future generations. Mitochondrial gene editing offers a different path. If we treat an adult patient's heart or muscles, that is called somatic editing. The changes are not passed to offspring. This is ethically similar to a kidney transplant or standard drug therapy, so it faces fewer regulatory hurdles. But if we use these base editors on embryos to correct the mutation, rather than replace the mitochondria with the donors, we re-enter the debate of designer babies. However, many proponents argue that fixing a typo in a gene is actually more natural and ethical than swapping the entire organelle with the donors. Looking ahead, the trajectory of mitochondrial medicine suggests a move toward total control over cellular energy. Researchers are already looking beyond rare diseases. We are talking about MitoPrime, exploring ways to bring the versatility of prime editing, which can insert or delete whole chunks of DNA into the mitochondria. But perhaps the most universal application is aging. Mitochondrial dysfunction is a hallmark of aging. It is linked to Alzheimer's, Parkinson's, and general frailty. As we get older, our mitochondrial DNA accumulates damage, and our energy production drops. In the future, rejuvenation therapies might involve editing mtDNA to restore youthful efficiency to the electron transport chain. Imagine a therapy that doesn't just cure a disease, but recharges the battery of life itself, potentially delaying the onset of age-related decline. To conclude, mitochondrial medicine has transformed from a field of palliative care where we managed symptoms with vitamins and hope, to a field of curative intent. By overcoming the biological barrier of the mitochondrial membrane, we have unlocked the ability to edit the source code of life's energy. As we move through 2025, we are witnessing the first steps of a journey that could eliminate an entire class of incurable genetic diseases. The body is an electric machine, and for the first time in history, we have the tools to fix the generator. Thanks for listening to G Fun Facts Online.